Well, good morning, everyone, and welcome in across Canada and beyond for another exciting Exploring by the Seat of Your Pants broadcast. So, my name is Jesse, and in my role, I have the great privilege of getting to connect with the coolest scientists and explorers on planet Earth and bring them straight live to classrooms all around this planet all school year long. But it's summer, so school's out of session, and we are here on a bonus broadcast with our friends at the CCGS Amundsen. So, the Amundsen science team are the best explorers of Canada's north and beyond that I could possibly think of. We have done a bevy of live broadcasts with them in the past, and they get the incredible job of getting to explore some of the most remote, amazing, untouched places on this planet. Now, when I was a boy, like I'm expecting many of you, when I wanted to learn something, I had to walk uphill both ways in the snow to go to the library to grab the one encyclopedia that might have the entry on what I want to know. And now we have the privilege of getting to go live on a ship in Canada's far north with a live feed from the ROV exploring the seafloor. And if that is not the coolest thing ever, I do not know what it is. And so today we are going to be joined on an epic live ROV journey. We're going to learn all about their mission, the ship, and more. Aidan and John are joining us in just a second. And what I want from you as an audience is to share all your questions in the chat. Anything you ever wanted to know, what they eat on board, how they sleep on board, how long they're out, how deep they're going, anything you're keen on, today is your day. And so, without further ado, you're not here for me live in some office somewhere. We're going to take you live to the ship. Aidan and John, thank you so much for joining us today, gentlemen, and tell us all about what you're up to.
Oh my God. Good morning, Jesse. It's nice to be here. Currently, Aidan and I are sitting outside the RV control room. So the RV control room is the place in the ship where we have all of the live information that's coming from the vehicle itself. For folks at home who are not familiar with an ROV, it's an underwater vehicle. Picture it like a giant underwater robot. It's cabled, tethered to the ship, so we get information from all the cameras that are on the ROV. All of the scientists and the pilots who fly the ROV are getting that information in real time. And what you're seeing in a home. And it allows us to explore these very unique, untouched habitats deep hundreds of meters under the surface of the ocean.
Super cool. And so this mission that you're on, how long are you actually out on the water? Is this like a 1 day thing, 1 week? Are you gone for 6 years before the mast? What's going on?
So first leg that we're on, which is the first leg of the 2020 expedition, we're on the water for 28 days.
And so you set off from where, and where do you end up ultimately on this journey?
Yeah, so we departed from Quebec City on the 10th of July, and we'll end up in Iqaluit, and that's kind of where we switch out with the next leg that'll continue a little farther north. Trade off all the different science teams, and yeah.
So as you go further and further north, I mean, it's summer up there too as well, of course, for anyone who might be joining in South America. And so is it icy? Do you have to break through the ice? Like, you are on an icebreaker, so if you run into stuff, you can plow through it. Is water clear when you're going up there this far north this time of the year?
We're kind of in an area known as Iceberg Alley. So there's no sea ice per se, but there's a lot of larger icebergs, which are pieces of glaciers from further up north, which are carried by ocean currents all the way down here along the coast of Labrador or the Nakivik. And so that's kind of where the conditions that we're in right now. We haven't actually seen a lot of icebergs today, thankfully, because sometimes I can make operations like deploying the ROV challenge. So we're happy not to have seen any icebergs today.
I was going to say, from the perspective of people that have watched way too much Titanic, like, icebergs are beautiful, but in an area where there's lots of them, at night, do you have lookouts? Do you have watches? Do you have people that are making sure that you're not putting the ship in danger and yourselves in harm's way?
Yeah, so there's on the bridge of the ship, there's someone at the helm, and there are multiple officers on watch, and they're keeping an eye out for icebergs. We get the kind of forecasts of the ice conditions from the Canadian Ice Service. Get some satellite imagery, so we have an idea looking ahead as to what the kind of conditions we're going to encounter and how to safely maneuver around it.
So tell me what we're seeing here, because we're on dive 78. We're live. This is amazing. You've done this all the time. Dive number 78 already in a 1-month voyage. So how long does it take the ROV to get to the seafloor? How deep are we? Like, give us a little bit of a sense of what we're watching here.
So I think we're 700 meters or so.
Yeah.
And so our dive started at 9 o'clock for us, so about an hour and a half ago. And we just touched the sea floor just a little bit before we started the live stream. So it took about an hour to get to the bottom. And it's much more entertaining now. Otherwise, it's just a lot of blue and a lot of that marine snow in the background that you still see now.
So where we are right now, this is an exploratory dive. This is an area known as the Makkovik Hanging Gardens. So picture it like a massive underwater cliff, and there are dense aggregations of cold water corals growing inverted upside down along the side of the cliff. And from the Amundsen, we've done a lot of exploration of this area generally to understand how the corals are distributed. But this is more of an exploratory dive to kind of test our understanding of what kind of conditions they live. So it's a scenario that we've never dove on before, but based on the steepness and the characteristics of this underwater cliff, we suspect that this could be the place where we're going to find these dense aggregations of corals. So we've kind of started at the base of this underwater cliff, which is hundreds of meters high.
Yeah.
And we're slowly making our way up the cliff, hopefully to encounter some of these dense corals.
You're just climbing a mountain underwater with a robot. And so cold water corals, I want to harp on this for a second because I'm a biologist by background. I read every nature book in the whole world ever when I was a boy, and I didn't even know cold water corals existed till I was quite a bit older. So, you're beyond the reach of the sun at this point. There's nothing. How does a coral survive this far down to begin with for anyone who's never heard of such a thing?
Yeah, so for folks that are tuning in at home, you may be familiar with tropical corals, the reef-building corals in the, the warmer parts of planet Earth. And the corals that we have in the Eastern Canadian Arctic, they're not. Most of them are not reef-forming, and like you say, Jesse, they're growing hundreds of meters under the surface of the ocean, beyond the reach of the light from the surface. So with tropical corals, they have these symbiotic algae that live in the tissues of their polyps. That's good. And that's how they produce their food. So because we don't have any light down here, the corals that are growing in habitats like the hanging gardens here in Makkovik are entirely dependent on all of the productivity, the food that is produced in the upper layers of the ocean and slowly sink to the deep sea. And basically,
oh, we lost your audio for a sec, John. Sorry.
Oh,
there you go. Oh, good.
These corals are in a kind of a prime location because, as I said, they're growing along a steep vertical cliff. So you can actually see in the imagery right now what looks like snow is actually just sinking organic material. Could be the fecal matter from zooplankton. It could be other, sinking phytoplankton or sea ice algae. And all of that material is just raining down slowly, and working on the side of the cliff in a prime location to just sift that out of the water column.
Yeah. I mean, you've hit upon two things here that are super cool. One is the fact that coral are animals, and a lot of people do not know this. Adults don't know this. I mean, they form this relationship where they get all the pretty colors, but at base, they're like an upside down jellyfish that anchors themselves to the seafloor. And two is this marine snow, which I cannot stress enough. If you are on a live dive with any ROV, if you're in a submersible, there is this constant rain of dead things and little things that are going down and founding these whole ecosystems in the seafloor, which is something that we know now. And I think a lot of people know, but it's science that is still really, really recent. Like, we did not know this in the 1930s. We had no conception that there were such amazing ecosystems on the seafloor. And so really, really neat stuff. I want to harp on this laser thing because this is one of my favorite things in deep sea dives. Why are you pointing two big green lasers at where you're going? Tell us more.
Yeah, so as you can kind of see the sea floor, you can see all these different kind of sea stars and anemones, but from just the video, you really don't have a good idea of how big they are. So these lasers are 10 centimeters apart. So they give us kind of a reference size. So those two dots are 10 centimeters, and that lets us extrapolate to everything's size. We can see all the super big corals, all the super teeny corals, and we can turn them on and off so we can get some pretty pictures, but we can also get the scale, what's going on down there.
I was just going to ask if you could turn them off. There you go. I've seen it where it's something on like a 6-gill shark and it's this 20-foot monster and it's this tiny little bit of it that you get against with the lasers. Very, very fun stuff. A quick query too, because we're starting to get some great questions in the chat. So if you are doing this scan, you're going up the mountain, you're checking things out. If you saw something like that fish that just darted into frame or something on the right, 10 feet away, could you stop and go and look at that now? Or is this meant to just be like a transect where you're going straight up and seeing a general breadth of what you see?
Yeah, so the ROV pilots are actually very, very skilled. It's amazing how much maneuverability they can get kind of 700 meters below us. So the ROV has like a robot arm on the side. So at any time we can kind of pause, adjust the thrusters for the current so we can try and stay as steady as possible. The ship itself has kind of a positioning system. So as the waves push the ship back and forth, we can stay in the same relative area. But we're all kind of scientists and the ROV technicians and pilots working together to go through and explore. So if we did see a super cool coral colony or a shark we wanted to try and chase down, pursue that for as much as we can as it kind of resume to our dive plan.
As a person who played endless amounts of video games as a boy, the idea that you could play with a little robotic arm and it could actually actuate something underneath the seafloor hundreds of meters below is like magic. One of the coolest things ever. Do you ever have big things come up and like lick the lens, or are most creatures kind of scared and running away from this giant weird robot with lasers under the water?
There's so it is 700 meters below the surface. So as we kind of talked about before, there's no sunlight down here. So normally it's pitch black, and as the ROV's kind of coming down, it's rumbling a bunch, motors are going, and that's producing vibrations in the water. So there's both the light, the vibrations, the sound of the ROV, and all of these animals can kind of detect those things. So sometimes they're avoiding us as much as possible. It's a very bright wake-up call for them. But they're also, some of them do kind of become more curious. And, you'll see some of those like fish swimming by. Maybe they're just trying to hide behind a rock and not get spotted by us. But
sometimes there's certain species.
Oh, John, sorry, pull the mic up a little closer. Sorry, buddy.
I was going to say, while some fish and other species that we see down here are deterred by the lights and lasers, some are attracted by it. So you might see redfish, for example, that are kind of following the lasers around like a cat would.
Cool. See, thank you for that analogy of my day. We could just have a whole byline in the future. Underwater cats, the redfish of the. Um, Jean-Marie has a bunch of questions, so thank you for those. Please keep coming. The more you ask questions, the more I will pass them along to our friends on the ship. Does ASTRID, they know that the ROV is called ASTRID, which means they're like well on board with everything you're doing. Does it have stereoscopic vision capabilities in any way? Like, how tailored can you make the sight with what you're looking through?
Sorry, your audio kind of out with the communication question.
That's okay. Yeah, does ASTRID have stereoscopic vision capabilities? Is our question.
At this point, no. There's multiple cameras on it, but it's not set up for that stereoscopic camera setup.
Fair enough. We've got another one for Jean-Marie. What's required to be an ROV pilot? So how do you get this amazing gig trying to chase sharks down and see cool things under the water?
Well, we have a couple of RV pilots right here.
Yes.
Are you able to answer a question, Gabby?
Come on in. The more the merrier. Well, hello.
Oh, I'm on
You're
the screen.
on. You're live.
Okay.
You're wonderful. Hello. Nice to meet you. Well, we've got people that are obsessed with you and want to know how someone gets your job because it's one of the coolest jobs in the whole planet. So how does someone become you? Yeah.
1 second. Yeah.
Better? Can you hear me now?
I was just having trouble. I'm just having trouble. Like I hear myself too much. Okay.
Yeah.
Okay.
Better?
Much better. Yeah. How do you get to be me? Well, we have a team of 5 out here, all working as ROV pilots and technicians. We all have different specializations, but the main theme is that we've all specialized in some form of technology. So we have a couple people who have studied robotics. We have a mechanical engineer. We have somebody who came up through the military, and he specialized in hydraulics in the military. There's a lot of different ways to get here. Did we lose him?
No, you're good.
Oh, okay, good.
Yeah.
There's a lot of different ways to get here. And the real thing is like, you end up following whatever interests you, because if you're doing what interests you, you end up doing these like crazy deep dives, following these rabbit holes, learning a ton more about something than any normal person would. And then you just keep following it. And my biggest advice, if you want to do ROVs, is just start building, start experimenting, get into whatever technology like really interests you. If you like making maps, like we have somebody here who trained in making maps underwater. Basically, just like follow your interests and like don't be afraid to go all the way.
It's amazing, given them. I've done 2,000 broadcasts with scientists and explorers the world over, and everyone's advice, whether they're a geologist, particle physicist, or ROV pilot, like be a nerd, keep it up, go enjoy some cool stuff, and you end up with like the coolest careers on the planet.
Yeah, exactly. You just gotta like give her like all the way.
Do you have time for another question? Because you're wildly charismatic, and I appreciate you immensely.
Well, thank you.
Okay.
Yeah, go for it.
Sweet. So you said rabbit holes, so I want to know, because we've got a whole bunch of questions about this specific dive, and that's very cool. We're going to get to those. But what's your dream? Like, what if you had complete carte blanche, you ran the ship, what would you go exploring? I want to know.
Okay. So I'm going to tell you what I actually am like passionate about doing, because for me, it's become over the years about building teams of people who like support each other and believe in each other, because those are the teams who can do really amazing things. So I think when I started, I wanted to find shipwrecks and I wanted to map things and I wanted to explore. And now it's become like I'm 19 years into this career, and it's become very much for me about like building teams that can do amazing things. Yep. So I love putting together, I've loved putting together this team with a lot of help from a lot of talented people. You know, that's what my goal is out here.
I can't stress enough. It's been really fun because we've had the chance to work with the Amazon a bunch of other deep sea research vessels all around the world. And universally, it's like a bunch of people that get the chance to work with an international team of collaborators, like the coolest, nerdiest scientists in a wide variety of fields that love the opportunity to go on big adventures, find new things. And so your statement has been echoed by a lot of really, really special people. And I just really appreciate you coming on. Bonus, unexpectedly today, to join us and share a little bit of your insight and passion. You rock.
Yeah, of course. Thank you so much, Jesse.
Thank you so much.
Okay.
If you got to go, we'll leave you to it, but we'll bring back John, or
Okay.
we'll wrangle the whole crew.
I'll go find him and send him back.
Wherever he is. Okay.
Yeah, thank you so much, Jesse.
Thank you so much. And we're back with Aiden. So speaking of the specific dive, Alec wants to know, is there anything specific you're hoping to find on this dive? Like, is there like a dream scenario that you'd have?
So kind of as John mentioned earlier, it is an exploratory dive, so we really don't know what we're going to find. We have lots of kind of diverse teams on the boat, so lots of people interested in lots of different things. Right now, kind of...
Oh, Aiden, bring up the mic again, buddy. Sorry. Here you are.
Yeah, so we have lots of different things.
I'm going to stop you one more time. I'm so sorry to do this. Your audio is just a little iffy. Let's smack it. It's very emotionally satisfying to smack the mic. Can you hear me now? Better? Maybe.
Let's see.
Now you're good.
Oh, we're good now?
Now you're good.
Beautiful.
Mwah.
All right. So, so I was mentioning, so this is an exploratory dive, as John mentioned earlier. So we really don't know what we're going to find down here. But yeah, based on the mapping and the region that we've explored previously, we're hoping to find kind of these deep sea cold water corals and really anything else.
If you like, Jesse, so we're just sitting outside the ROV control room right now to facilitate the questions, but we could have the kind of lead scientists on this dive describe their objective directly too.
1,000 times yes, John. We always want lead scientists. We want ROV pilots. We want like someone in the bilge pump. We want the chef. Like, bring on everybody. This is the joy of being live at sea.
Okay, great. Well, we have a mic in the room, and I'll get the chief scientist to jump on and kind of describe what their main objectives on the dive.
Yeah. Outstanding. Well, while you're doing that, Aiden, I'm going to share another one for you, which is from Nate. So Nate wants to know, have there been any noticeable changes to the seafloor compared to the last time you guys checked this out? So you're doing so many dives every journey, but even between missions any given year, do you notice different things? Is there anything weird that you notice when you're down there?
So yeah, with this specific dive, as we mentioned, we really don't know what was down here before. But with previous ones, it's pretty kind of stagnant area, but sometimes there might be kind of trawling marks that we see, or new colonies, or kind of new populations. But I think our chief scientist is ready to describe their
We'll come back to it. We got all the time in the world. Hello.
Yes, I will watch. I have too much. Yeah. Okay. Hi.
Hi. How you doing?
Yes. Yes.
Got a bit of audio. Cutting out a little bit. This is the joy of live broadcasting. Something should go a little wrong. Otherwise, it's no fun.
This is a new site. We have been in it before. We know that there is abundant corals living in here. It is a very interesting area from a seafloor biodiversity point of view. And so today we're exploring a new site that, based on some of the data we have, we should be seeing some very interesting features on the seafloor and corals. So we, right now, we are transitioning from a soft bottom to a cliff. This is a portion of the dive we've been waiting for. We don't know exactly what we're going to see. We're looking for corals growing on these walls, these cliffs. At the beginning of the dive, maybe some of you saw it, we had a coral on the seafloor. It looked like it had fell from the wall. So they should be here, but we have not seen them yet. So the idea is that we would like, one, to confirm that they're present, so that it's going to help us to understand if the models we have been working with are actually true. So to validate those models, but also we will collect samples. We have a lot of different people interested in this area and studying these corals, and some of the scientists here can talk a little bit soon. So we're going to sample some coral, some sea stars,
Yeah.
in this area. So we are at a steep portion of the dive, but we still have coral, so we don't know what to expect. Is it about 600 meters now?
Wow.
And we started at 700, so we've done 100 meters so far. We're
So
seeing
much.
a lot of sponges, more than before, right, guys? So this is a sponge called Makkovik, and maybe later some of you can talk a little about the sponges and the evolution of the sea stars. It looks like stars are hidden there.
Yeah. I want to query this because we've talked about the fact that this is an exploratory
So we
dive.
have 8 people moving room right now. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 people. We have 3 pilots taking care of the ROV, and we have how many again? 6 scientists that are taking photos of what we see, and taking notes, making observations about the fauna on the seafloor. But the actual analysis of all the video and the data getting out of it comes much later. So we're just making some observations, but later we're going to really spend the time analyzing everything in detail. So we want to know, the different species that are living in here, how many of them, where they are in terms of the depth of the water column, for example. So some of them prefer to be at different depths. And we know that the water temperature here right now is 4.3 degrees Celsius. So the ROV has a CTD, so an instrument that can measure bottom temperature and salinity as well. Oh, just saw a squid. Yeah, so
Cool.
I think we just went through that very steep portion, and now we've seen a few rocks and outcrops, but not yet our corals. So we'll see.
We'll see. Fingers crossed.
So like I said, if we are close now, we are 586 meters. This cliff goes all the way to 260, I'll say 5. Yes, so confirmed. And it means that we still have a while to go here. Our objectives, they can be adjusted during a dive. So depending on what we see, we might decide to stop and begin sampling and collecting animals. Oh, wait a minute. That is a coral. That coral. No. Bottom left. Yeah, we just went by it. Coral.
Coral.
There is a coral on the top of the wall. We found it. But just one second. Should we make a waypoint for that? Oh my gosh, it's a big one. Should we make a waypoint for that coral in the first Starboard? You guys can think of a
Look at
name
that.
for this waypoint?
There it is.
There's this. Yes, okay. This is a large coral. As you can see, we call this, the scientific name is Paragorgia. And we call it the bubblegum coral too. It's the common name. So this coral, they can grow to meters in height. They are very, long-lived and very important habitat for a lot of species. You can see right now we're zooming in on it, and it has a lot of. It looks like it's sediment on it, but it's looking, isn't it, guys? Like, look at the base of it. It's very. So a coral like this, you're gonna see the skeleton, or the branch, the base and the branches, and then you're going to see the polyps, which are the portions of the tentacles in a little mouth, and they feed and take care of the colony. We call a coral like it's a colony because it has 7 polyps, but the branches are really thick and full, right? Anybody have thoughts on that? Or am I just being
I love it. How lucky for us to get to be live as you make that big discovery while you're talking to us.
So, yeah, which is because yes, yeah. Oh my God, yes. There is a escape on the eyes. Look at the bottom of the screen, and Simon is zooming in on them now. You're gonna see the eyes. So that's the easiest way to identify the shrimp. So you see like 1, 2, 3, 4. There is yes, and the just remember that it was 10 centimeters apart, the 2 dots you see on the screen, right? So give you an idea of the size. Stop the little polyps open. The little polyps, right? They are the polyps and the animals, right? And it's interesting when you put this polyps always have 8 tentacles. No more polyps.
Oh. Cool.
Potentially dying, but we saw the size of it. Anybody want to try to imagine this and get an idea? Maybe you can see the color here. Colony. So it means that it's probably been living here for a number of years. I think.
Cool.
And if it's dying now, maybe there was some sort of event. It's falling into the sediment, right? Where more sediment started falling on it for whatever reason. No, right? So I wanted over a meter. So we think, oh, you can see the shrimp still. Amazing. You see?
It's like a little tropical rainforest underwater. I love it.
This is amazing.
It is.
So I'll just say a few words in French. Donc, on fait une plongée maintenant à Makkovik. On est en train de regarder pour vous, vraiment de proche. La plongée, on va souvent parler en anglais, mais si vous avez des questions en français, vous pouvez laisser un message sur le chat. Puis on peut vous parler un peu en français. Donc, on voit maintenant, on fait des zooms sur les fonds. Et aussi, on voit qu'il fait tout on voit qu'il est en train de pousser sur les corails. C'est des hydroïdes, en France. Hydroïdes. Je pense que c'est ça. En tout cas, on va recommencer en anglais, mais si vous avez des questions, soyez à l'aise, et on va répondre. It's for the audience.
It's such a beautiful thing to get to check out.
Captain, do you see don't you think those are the gulls? Or they are just really retracted? They go through earlier. Yeah, okay. You don't look at them. Right, yeah. So you guys see they do look like they're flowers. I think it's not, it cannot be all this there.
Were there any for the chat or Jesse that we can bring to the room for you?
So just free flag coral.
Oh, sorry, what was that, John, too?
I just wanted to see if there was any questions from the chat.
Oh, yeah.
Do you have any questions that we could bring into the room for you?
We certainly have a bunch. We have people talking about the nature of coral. We've got shrimp eyes looking back. We've got people asking about the autonomy of ASTRID, how much it can move around.
But we're not seeing any here.
Doing robotics.
Look at the detail
a
of
whole
this
bunch of questions that I'll be leaving you two in a minute. But it's just such a privilege to get to be here for the first 30 minutes of this, and the fact that we found something so cool at the end of my little section here is great. But I hope you guys get the best audio and get to explore more and get some extra zoom in on this. And you can check out everything on the right with Veronique bringing stuff up if you need to. Enjoy the dive, guys.
So you guys.
Thanks for hosting us,
to move
Jess.
on, or you wanna
Enjoy, I hope you find the coolest stuff ever. Spanish
So we
galleon,
have
epic new sharks,
that's okay.
lost plesiosaurs of the deep,
No.
whatever you guys are looking for, and more with this unbelievable coral. We've got some really great zooming in on now. So I'll leave you guys to it. Have fun.
Thanks,
Another
Jesse.
thing interesting about this is that this is the first live coral we saw in this dive, but we're looking for this one and another species. The other one is the one that we see in high densities. So this is a great start, but we want the other ones too. The other one was a Prenoa. Yeah, that one is the popcorn coral, and we're going to talk more about it soon. Okay, so these white things you see on the cliff, these are sponges. So sponges are animals. They are some of the simplest forms of animals that exist in the planet. They do filter feed, so whatever is they're eating is coming from the water, and they have little cells that help them to get everything that they need from the water. So they will feed on different things. They have a lot of bacteria and a microbiota associated with them. Why I don't see it? Second. Oh yes, I see a basket star. Yes, yeah. So this, this is what these sponges are. They're called Plicatelopsis. We don't know much about their ecology. We don't know much about how old they are. But look at that one, Wilder. The one, the top one, has no holes at all, which is very interesting. So you might notice that some of these sponges have holes on them, but we just looked at one here to the left of the screen that doesn't have any. And, we are not sure what the holes mean, what they are, where they're coming from. But there is a study that a team that is here with us is doing that is indicating that they are fed by those sea stars that you see around them, so that the holes could potentially mean that. Do you want to say anything about that? I'm going to give you the mic. Maybe it's going to be...
Yeah, so my name is Wilder Greenman. It's my second year working on the ROV here. And so our study last year started when we saw a lot of the sea stars that you see there in the frame. They were hanging out quite close to the sponges. And like Barbara was saying, it looked like there might have been bite marks in them. So we kind of wanted to investigate if we could find more evidence that the sea stars were eating the sponges. So we took some samples that had been collected, and visually we thought that there was a relationship there. So we did a spatial analysis, and it looked like, yes, the sea stars were clustered around the sponges. We also looked at the isotopes of them. So with looking at the tissue sample, we did geochemistry back at our lab at Dalhousie University. And yeah, it seemed as though there was more evidence that the sea stars are eating the sponges. So it's this kind of interesting new evidence that there's kind of this distinct food web where the sponges are feeding some of the other sea stars and things around them.
And regarding the question about the microbial diversity,
and then if you pay attention, you're going to see that the sponges are common and the sea stars are common through the dive. So they do coexist and they have a relationship.
So regarding that last question, so when we grab the sponges and take samples, that's really our big
opportunity. To say something about that and then pass it on to him? He's talking.
Yeah, so the ROV is able to take samples when we're down there. So if they see a colony or different curls that they're kind of interested in looking at, then we're able to kind of take a little sample, bring it up with the ROV, and then do a whole bunch of science on it. So we can sequence the DNA, the RNA, and try and get an understanding of the kind of microbiome community, because just like how we have a microbiome in our gut that helps us digest our food and stay healthy, so do all other animals. So these ones, instead of using algae like corals and sponges in kind of photic zones that are able to see the sun, these ones have different communities that help them survive down there.
So I think it's really specific, right? We need to go down and explore. What is that? Like, is that Primnoid skeleton right there? Like, just pass by? No. Oh, another one there. See, oh, there's Primnoid there, isn't it? Like at the top left, yeah, we say there, right? Top, middle. It's a dead one. It is dead. Yeah, so, oh, it's a graveyard. It's lots of dead ones. Okay, so this is... Is this, are both dead? No, these ones are alive and attached, right? That is so interesting. It looks like they're dead, so they are falling. Yeah. Yes, where did they fall off from? So, okay, yeah.
So is there another? No, it's just that.
Oh, okay.
Yes, they were bringing up kind of the RVs.
And it's
Okay. Is that the question?
Yes.
Okay, so the question is, what's the autonomy of ASTRID? Dive duration, depth, distance from the ship. So I wouldn't call ASTRID autonomous by any measure. ASTRID is essentially a tractor. We fly ASTRID around with full control from the surface. ASTRID has a tether connecting it to the ship, and all of the controls for ASTRID are on the ship. So ASTRID can go approximately 100 to 200 meters away from the ship, and to a depth of, in its current configuration, I would say 1,700 meters. The duration of the dive could be pretty long. We tend to structure our dives aboard the Amundsen as 8-hour dives, but in theory, an ROV can stay down for a very long time. The limitation for science work is typically sample payload. So we take all these samples on board, and at some point the ROV gets full, and then we have to come up. But otherwise, ROVs can stay down essentially as long as you want them to, days, weeks. We just keep swapping out pilots. You think that answers the question? Cool.
Oh yeah! Oh my gosh! Can you zoom in on that? Yes. No, it's there. Right. Yeah, it's there. I see it a lot. That is so cool! Wow.
Yeah, so kind of as it's going, we're able to take all of these latitude, longitude coordinates. So we're able to really lock down these very specific parts.
Can I talk?
The ocean's huge.
Yeah, so right now, this is the second bubblegum coral that we see, and we're trying to zoom in on one of these parasites that I had mentioned. So you might see right in the middle of the screen, there's two long wormy-like things, and they actually are little crustaceans. So they live inside of the coral, and I think this might be the first time ever that somebody sees them alive, because they're so difficult to observe alive, pretty much. So are we at the maximum of the zoom we can do on them? All right. I think you're seeing the copepod. So yeah, the other one, yeah, but it's just weird thing. Let's see. Yeah, it's hard. It's, yeah. So I don't know if the audience can hear, but we're discussing in the room whether those are the parasites, if there's something else. I don't think they're part of the coral. They look very different from anything that you would expect on this coral, but it could be. It's because we know that they have these parasites that we're kind of thinking that's what it is. What are these little orangey things? They're polyps, right? Yeah. Okay. I think I'm satisfied with this one. Again, we see portion of the column. This looks really old. And how old? I would say a few hundred years, but we don't know for sure. This is a coral a little bit difficult to age because of the type of skeleton they have. But like I mentioned, they grow very, very tall. And for this one, we're going to try to give you guys a number. And then is that me? Oh, coral prinoa. All right. Finally. Okay. So now we're happy. We were happy before too. The bubble gums are nice, but we were looking for these coral gardens, right? And they're mostly made of these species that you see right now on the screen. That is the popcorn coral prinoa. And there is a little juvenile there. So put a waypoint on that. We have a student here, Catherine, who is very interested in studying the juvenile corals. Do you want to talk a little bit about your project? Let me give you the mic so it's better.
Would you say that we're making a waypoint for the juveniles? Yeah, I was referring. Thank you, Laura. Hi, my name is Catherine. I'm a PhD student at Dalhousie University. And for one of my projects, we're going to be collecting some of the juvenile primnoas. And we're going to be looking at their growth rates and comparing them to the early years of the older colonies. And then also looking at how things may have been changing over time in terms of their diet and their growth. And so in the middle of the screen, you can see a baby primnoa and then an older colony, probably 20 to 30 years old, on the right of the screen. Now another primnoa with a Plicatolopsis sponge. And do we know what the mass is? Are they just, do you think they're worms?
Yes. So 2 worms. I think that's something to come back here to sample. We would try to collect some of that. Might be hard, but that's
Lots of encrusting sponges. That's the white stuff that you see on the wall.
there's interesting because you're interested in the baby. There's a few juveniles here, right? And we're seeing some dead on the seafloor. So maybe this is new recruitment, right? Like,
A little crinoid on the sponge at the top of the screen. They feed on what's in the water.
and again, notice these sponges don't have the holes on them. So I think this is very interesting because it's telling us that the holes are not a characteristic of the species. Otherwise, all of them would have them, right? So I think the idea that so we're seeing a few more primnoa now. You can see that.
Clark, can we make another baby primnoa?
Oh, yeah.
Look, there's lots of them. And a young Pyragorgia.
Yeah, lots of holes on that. Did a shrimp on it?
What is the wiggly thing?
Worm.
Yes.
Yes, it's a Pyragorgia. Yes.
So on the right of the screen, you can see a smaller primnoa and right above it, a smaller Paragorgia. So the 2 types of corals we have here so far.
It's right where your bubblegum coral was. It looks like the anemone's growing on a shell.
Some of them do that.
Yeah, look at that. Just roll like a rough. Has been. So this is interesting. we are very excited and happy that we just saw, all of these corals because that's what we're looking for. So it tells us that the models we're using to identify their presence, they work. We knew that, but every time that we validate that is obviously very important. Now, one difference is that we have not seen the high densities yet, right? We only saw a few of them, and we wonder if there is portions of the wall here where you would see lots of them versus just the few sparse ones. There is a lot of sediment in here. You can see how the bedrock is covered with sediment, and that might have an influence on it. Now we are at 553 meters. For you guys. So just so the audience also knows, in the ship, like I mentioned, there's a few people in the OV room, but we also display the dive through the ship, different screens, so people in the ship can watch just like you're watching at home. And then we always have visitors come and have a look, talk to us about what we're seeing, because there is there's a lot of diversity in terms of expertise in the ship. So the people in the room are really interested in coral, for example, right? But there's a lot of people that are studying seawater or sediment and different things about those. And then they will come and be very interested in what it is that we're doing and why.
Does that see start on this one? One in the middle?
Oh, in receipt, yes. I guess close this.
Now we're at a very steep part.
Yeah, so I find it very interesting how some of this, the species we see here, it's very specific. There is a lot of species that we can't see from afar that we would need to do a close up to actually see. So it's not fair to them, but in general, we are seeing mostly the same, right? We're seeing these white sponges, the cataloxes, we see the sea stars that feed on them a lot. We see our corals, even though we're not seeing many of them yet. So it's very, very niche environment. So I guess the species we see here really like this spot for some reason. And I think it's one of the things we're trying to understand is what is so special about this site for this species. But look at that one in the middle. You see that? Wow, that's so strange. Yeah, one big bite. The shrimp again on this orange.
That's sea anemone at the top.
Yeah, that's a sea anemone. It looks very different from the other sea anemones because you're not seeing the tentacles, right? So the tentacles are all contracted inside of the anemone, so it just looks like a little ball. But once the anemone is relaxed, it would have all the tentacles out. And it could be that it's not happy because we're so close to it and there's some sediment in the water, but soon we'll come back to normal and open tentacles and do what anemones do. Yeah. Could we do a close up of that? Dave, could you point for Simon? So we're going to do a close up to see if the sea star is on top of the stone. It's not the same specimen.
And for this leg, it sounds like we have about 12 dives planned.
Beige, type
So
of sponge
for
is
at
beige.
this site,
Definitely
then we'll go a little bit farther
doing
north
something.
for at their
The
site,
stars,
and then
they,
more
they
north,
are beautiful.
another core.
And they like corals and sponges and a lot of other animals. So you might ask, what kind of animals feed on corals or sponges, for example? And one of them is sea stars. They like them. And so I don't think it's unusual that we'll see something like this. They're going to be wrapping the whole coral, and they do have a structure that they use to feed on these guys.
Some brittle stars too, the little orange arms that are coming out of the sponge. They're another type of sea star.
And so that is also interesting. Is each time that you do a close up, that you zoom in the specimens, the animals, you're gonna see more, right? So we could not see from afar all of those little stars. We call them brittle stars, with these very long arms you see in there. But because we're doing a close up, we can. And it's very interesting. Okay, you guys happy? Can we move on? Move on then. Could you put the lasers back on, please? Oh, they're hidden. Hidden because you zoomed in. Yes. That was funny. It's on. Sorry. Okay, so one of the questions from the audience was about, we have an idea of how extensive these coral gardens are in the region. And that is still a difficult question to answer. We know that we're talking about, at least dozens of square kilometers of the area where we do see them. Now that we are able to use multi-beam to map these cliffs, and now that we understand that these corals do like the cliffs, we're trying to make this association between finding the cliffs on the multi-beam on the maps and suggesting the presence of corals. Just saw another one at the top of the screen, top right. Nice. Yeah, so yesterday night, for example, we did more of the multi-beam survey, and very close to where we are right now, and we know that this feature, this bedrock feature, extends more towards the east. So I think we're gonna gather a little bit more data on the distribution of these cliffs through the expedition, through the next days, and hopefully we're gonna be able to answer that question very soon. Is that a juvenile, Catherine, for you?
No, I was looking at the sponge right beside it.
Yeah.
There's like a little window, maybe. I think that was too big.
So we're calling this our discovery dive because it's a site that we have not seen before. By the way, we are all seeing this together. So you guys at home, us in the ship, this is the first time that all of us are seeing this. So we think this is very neat, very exciting. And so we selected this site because, again, we saw those features on the mapping on the multi-beam. And then we decided to come and explore and see if we could validate that. But we also have other dives planned where we're going to go back to a site where we know those corals are present in high abundances, so that we can do better sampling as well. So it's a mix of sampling dives and dives where we are exploring to confirm our hypothesis of coral distribution. So it Can you get a sense of which depths we have been seeing there? Now we are at 527. So the first one very deep was the depth. $590. Okay. So since 590 years, we have been seeing them alive, and now 525. This that that's nice. Middle.
No, I can't. No, it's just far. You think? No, it's under it.
Oh, that signal again. So this is interesting that we see all of this stuff.
Not very big. It doesn't look
Not very big, very thin skeleton. Right in the middle of the screen where you see the lasers. That's another dead Prinoa. How do we know that? Oh, there's another one. Yeah. So we know that, well, for one, we don't have a lot of different species here. So we're seeing the bubblegum coral and Prinoa, popcorn. And we know that a dead Prinoa looks like that. It's a very thin, actually very smooth type of skeleton once the animal is dead. This is interesting. Now we saw a little cluster of them. Oh, look at that. This is a hanging garden. Yeah. Oh, look at that, guys. At 520 meters, we see this little cluster of them. Okay, let's explore this a little bit. Amazing. Oh, right. And I actually just looked. This site is at about 20 kilometers from the other site we know. So we're trying to also look. Yeah. Yes, it is. Well, let's make a waypoint, right? So waypoint here. So this is right now we're talking about a sea star. Is another type of sea star that we're interested in sampling. Again, as part of a food web study to understand what is happening, who is eating what in this area. There are deep water sea stars. Sorry about that. Actually, yeah. This is nice, right, guys? Got a nice little spot. Yeah, very nice outcrop. Beautiful. One of those you want?
The one in the middle.
Yeah. Did you ask Laura to make a waypoint for that too? So again, this is why we call the site the hanging gardens. We're seeing these corals hanging down, right, on the cliff. We are very happy. How much time we have? So we still need to be on deck by 4. Say it again. Okay, all right. Okay, he was saying 11:30. I thought we need to be back at 11. Yeah, that's right. So we need to be back on deck by 4, which means that we will be leaving maybe 3. Just kind of thinking out loud. And again, for the audience at home, it takes a while for the ROV to come down to this sort of depth. You guys come down at 20 meters per minute, more or less. Yeah, 15, 20. So it is slow to come back here. So when we plan for dive like this, we need to consider how much time it takes to come down and up. And there is lots of coral here. That is really cool. Happy? creatures. Yeah, so so if you look, like we are very focused on the corals on the wall and all the sponges, but if you do look around, you're gonna see lots of things in the water column. You're gonna see these little crustaceans that are flying in front of the skin. And wait, just 1 second, sorry. I'm just adjusting the mic. I guess I'll just hold it right. Yeah, so you're seeing all of the zooplankton in the water column. Sometimes you see lots of them, sometimes you don't see a lot of them. Well, it is telling us that, you know, there is productivity in this area, right? So there is actually lots of them in here. As we get far from the wall, I think it's just easier to see them. What is the relationship between them and the corals and the sponges? Well, we don't know that. The corals, they do, like I mentioned, they have tentacles and they can feed on, you know, small and a little bit of larger things. So, you know, maybe the corals like this place so much because there is so much food in the water, or maybe, you know, there is a lot of food in the water because the corals are here too, and it creates a productive environment. So, what do you know, it is a productive area. Do you want to say something?
Hi, this is Dave Cote. I'm a research scientist with Fisheries and Oceans too, and we just wanted to add that these plankton are migrating up and down in the water column in the daytime and the nighttime. So in the daytime, they usually go down at depth around the depths we're at now, and we can see them in fish finders as a band around 500 meters where we're at. And in the nighttime, they migrate up near the surface where they'll feed on the phytoplankton. So right now we're in an area that's too dark for phytoplankton, so they're just hanging out here away from predators, and then they move up the water column in the nighttime to feed.
Yeah, they're looking healthy, which is interesting because, like we mentioned, we saw some of the dead ones at the base, and we were curious about it. But they look pretty large, right? And thick, bushy, and healthy, and beautiful. Oh, look at that one! Look how thick. Yeah, can we do a close up of this large one on the left side? Yeah, close to the laser, Simon, please. Actually, I was supposed to be doing close-ups for the shrimp, right? But anyways, like we saw in the bubblegum coral, you know, all the shrimp on them. We know that Primnoa often has shrimp living on them as well. Now, it's difficult to study that when we collect a sample, for example, because the shrimp, they evade from the colony. So when we can see them alive, that is, you know, a great opportunity to spot them. So let's see if anybody can find a shrimp on this coral. I haven't yet. And usually we're going to look for the eyes, but we're not seeing it yet. It's beautiful. We're doing a little bit of a zoom to have a look again at the polyps hanging here. Hopefully you guys can notice how different they look from the other coral, the bubblegum, right? These ones really have the little polyps like all looking down. Yeah, sometimes this happens when we get too close to the wall and with the movement of the ROV, get some sediment. But I think that's good. We can move on. I'm gonna find some shrimp later. Yeah, this is another one. It's amazing. So we were talking about zooplankton, and they mentioned the vertical migration of the zooplankton. So we, in the expedition, we also sample zooplankton. So not with the ROV, now we're doing our visual survey. But actually, after the dive, we're going to put some nets in the water and we're going to survey the water, looking for zooplankton even at different depths and different layers along the cliff. So there's a lot going on, and we're also sampling sediment. We're interested in seeing if the fauna that lives in the sediment at the bottom of the wall is a little bit different if you have corals or not. So we have a lot of questions. That's why we keep coming back to Makkovik now. So we all know of the place now, and we're coming back with these more specific objectives and projects. Okay. So somebody was asking about what the corals are feeding on. There's a few of us that are interested in that in here, so we can talk a little bit about it. Yeah, so a coral is like what you're seeing on the screen now. We call each one of these a colony. And a colony is made of, hundreds and sometimes thousands of little individuals that look like little anemones, and each one of them has tentacles and have a mouth. Differently from the corals that you see in deep water, in shallow water. Oh, there is a redfish. Oh yeah, people would like to see that. And I'll get back to the feeding soon. Yeah. So we also seen a crinoid on Primno, and that's what is interesting about this species too, is that we, again, often the animals we see living on them, they're mobile. So if we collect them, let's say, using a trowel as a type of scientific equipment, we are going to miss on that because they live. So seeing them alive is very important for us to understand this type of relationships and behavior and all of that. Did you see a baby primrose that was just under this one? Yeah. Oh, that's nice. That's a crinoid. Crinoids are in the same group as sea stars. Just look a little bit different. There's a shrimp. Oh, guys, can you see the shrimp? You can see the antenna, like at the bottom right, more or less. Of yeah, you can see it. Everybody spot the shrimp. Oh, the crinoid is beautiful. And you can see now, I was talking about, how they feed and what they look like. Each one of these look like little flowers is a polyp that can feed independently. But they all feed for the colony. So that's what makes a colony. They are not independent individuals. They all work together. More shrimp. I saw another shrimp. Right, so we're talking about what they feed on. And I was saying that these are cold water, deep water corals, so they do not have the association with the algae that is in tropical corals, right? In tropical corals, that algae is going to give a lot of nutrients to the coral. But in here, they rely completely on what they're getting from the water column. And from what we know about them, they feed on a lot of very tiny things, on particulate matter in the water, but also on some of the zooplankton that you see flying around. Probably the smaller ones, like copepods, that sort of thing. And there's different ways that we know that. And we're going to talk more about it.
This is Wilder again. Yeah, so to add to that, so the corals are probably eating something kind of similar to the zooplankton. Stuff that's formed in the surface oceans in the upper kind of 20 meters. The difference is that the zooplankton can migrate vertically to go get it, whereas the corals have to sit, here at the site. They can't move, and they have to rely on that food to sink. Here now we're at 500 meters, so that's quite a long way for that food to come. So it's a delicate balance of waiting for that food to come to them. And if you look at their feeding structures, it's very small. when we come and look at them, we only get this very short window, so it's very hard to actually watch what they're eating. So in our lab, we rely on using isotopes to try and get an average kind of what they're eating throughout the year. And then what we can tell is that they seem to like quite fresh, particular organic matter, so stuff that forms phytoplankton, maybe some dead zooplankton and fecal pellets, things that form in the surface ocean that sink relatively quickly, and they seem to incorporate that. So yeah, we're kind of combining the different techniques that we can, isotopes, and also just taking the chances that we can to come look at them and see. We also use radiocarbon, and we can tell that food is modern, so we can tell that it's sinking quite quickly. And again, they're very fresh foods.
So, like I mentioned, we're now collecting the video and we're going to collect samples. And then when we get back home, we're going to need to analyze all of it. So the video analysis is an important part of what we do with ROV videos now. So Rylan can talk a little bit about it.
Yeah, thanks. Hi, my name is Rylan Command. I'm a biologist with Fisheries and Oceans. I work with Barbara on the footage and the things. Yeah, the actual analysis of all the video that we get is incredibly time consuming. It basically involves watching every frame of the video and drawing little boxes around all the animals that you can see. And doing all this manually takes an incredibly long time. Too long, actually. It's impossible to keep up with all of the footage that we're collecting every year. So something that we've begun to do is to integrate artificial intelligence models that learn the features of the animals that we're interested in, and it actually learns how to draw the boxes around the animals for us. So we can spend more time identifying fauna and thinking about ecological patterns and biological patterns rather than drawing all these tedious boxes. So it's made a huge difference in terms of how long it takes us to analyze videos. It's taken our workload for one cruise potentially taking up to 3 or 4 months to analyze down to a few weeks. So it makes a really big difference. Enables us to get really good data much faster.
Can I ask you guys, just out of curiosity, like on the wall, how are we surveying it? Like we're going a little bit laterally? You following that? Okay, sounds good. Yay! Yeah. Yeah, good question. So welcome our colleagues that are watching the video in name. Happy that you guys are tuning in. So the question was why the corals are hanging down. That's a very good question. So, and I don't know if I have a good answer. We know about this coral, right, Prinoa, actually growing attached to boulders. That's how, we've been known them in this region at least. So in boulders, on boulders, they would just attach to the boulder and, just grow in an upward direction. What do you say? Just growing like a tree, yeah. But if you think about it, then you have a bedrock wall like this. I think the angle would just make it difficult. So maybe they just grow like this naturally. They begin growing, pointing down. Anybody has any other theories about it? I think so. Yeah. So that is how they would just grow. But then the other question is there an advantage to it, right? And so we think that them in these positions, they pretty much save on energy. There's food being delivered to them more easily. Yeah, so another thing that is interesting about this site is the corals growing this way, if you think about it, if they can grow attached to a boulder and then grow up, you know, like a tree, versus if they're growing looking down, you would expect that the feeding structures, the polyps, would potentially look different. So in the ones that we see on the wall, they're looking down, and in the ones we see on the boulders, they're looking up. So, but this is something that we never really thought about until we found about this Makkovik hanging garden. So our colleague Vonda Hayes, the North Vonda, is listening. Vonda likes talking about this, how, you know, long time ago when she first started talking to Wilfred Bartlett, who was a fish harvester that was fishing in this area for a long time, he was showing her samples of corals that looked a little bit different. So they had the polyps looking down instead of up, and she thought that was really interesting. But, you know, we had no idea that there was a cliff, and that's the reason why they're growing in a different direction. So now we understand why. They're looking down, not up. And now we see less of them. And now we are at 495 meters, 495 meters. So we saw them, a lot of them in what kind of depth band? There was in the same area, right? What was it? 590 to 500 meters. That's where we saw most of them at this site so far. Isn't it interesting? Now we see all of this growing on the rocks and you don't see them. Could that be competition for space? There is so much growing on it that you just, you know, the coral larvae that need to settle on a hard surface to begin growing, they just don't find space. It's a pycnogonid. Yeah, close to the lasers. Yeah, it's a sea spider. Yeah, I just went by it. And we've seen a bunch of what looks like encrusting sponges. What is that? No, that is a clam. Can you have a look at that? Dave, can you point to it for a photo? Bottom, on the bottom, bottom middle. That, yeah, that round thing. Yeah, sorry. I think we're. Can we have a look at that? Because I think it's a clam, and that would be an important one. Could see the little tentacles. I'm wondering if that could be a clam. So for you guys, the audience listening to, we're gonna do 10 more minutes of the audio, and then we're gonna stop the audio and just leave you guys with the video. But I think the chat will be open. It had little tentacles coming out of it. It's just a sea anemone. Do you see like you can see from here? Yeah. Yeah. Mm-hmm. Okay. Can we zoom in on that? Yes. Thank you. We're trying to do a close-up of this thing that could be just a sea anemone. Not just a sea anemone, a sea anemone. Yeah, so you guys see also at the bottom of this outcrop, there's some white little things, and we think this is another type of coral. We're going to try to zoom in and have a look later. All right, so is this an anemone just with, what is that attached to them? Little crustaceans, oh yeah, okay. And can we do a close-up? Dave, could you point to these little polyps you see on the top, the bottom left? Bottom left. Yeah, this little group of, yeah, white. Sorry. Oh, do you see? It is a hydroid. See? See? The things we were asking what it is. You see the top of it. You can see the polyps. Now I'm talking about the, yeah, the brown things attached. Yes? Yes. So we know of primnoa growing on walls like this in other areas of the world. We know of them in Canada, at least in Nova Scotia. We know of them in the Flemish Cap area too. We know of them in Norway. Actually, in Norway, they can get in very, very shallow water. I've seen images of them in scuba diving depths, which is really interesting. We know of another species of the same genus Primnoa in the Pacific coast in Alaska, also in scuba diving depths. So we do see them on cliffs in other areas. I think along, Labrador, that was the first time that we saw them growing like this. We only knew of Primnoa growing on boulders, and our knowledge came mostly from northern Labrador, Hutton Basin area, where they're also very abundant, but again, growing on boulders. So, if you don't have abundant boulders, you're not going to see, high densities like we see here. So this is really special. Are we trying to zoom in? The sponge. The incrusted? Oh yeah, maybe it's not a sponge, maybe this is an ascidian. So yeah, it is, you might wonder about a lot of the things we see on the seafloor and how we know what is what. Some of them we know because we collected them and then we recognize them, right? Others can be really confusing and sometimes we don't know for sure. So we're zooming in now on what we thought could be a sponge, but now I think it's a different type of animal that we call ascidian or sea skirts. So sometimes we do need to zoom in to really understand. And a lot of things we really don't know. A lot of things we have no idea what is the species. We might know in general, it's a sea anemone, but we don't know what the species is. Until we collect them and have, the experts on the different groups identifying them for us, it's difficult. So these types of survey yield a lot of data. It is a chiton, yes. Yeah, and we can see, you might have noticed all these brown branchy things. One of them we just saw when we did a close-up. And at some point we didn't know what they are, but now it's clear that they actually are a type of animal that we call a hydroid. And we know that because we did the close-up and we could see the little polyp, just like a coral, but it's a little bit different. Oh yeah, can we have a look at that? Oh, no, I think we missed it. Yeah, so we need to think a little bit of time management to see how far we want to keep going. I just think it's interesting to know if we're going to see them shallower. But maybe we don't want to go all the way to 200 meters, right? So that we have time to come back and do some sampling. Is that oh no, it's a sea spot. Okay. True. Yeah. I think they're all ascidians. Oh, I didn't see tentacles. Yeah. No, yeah. See cucumbers. I'm not seeing them. Right, so this is what we're still trying to understand, this pattern. Why we see the corals in some zones, then we stop seeing them completely. And it's possible that we're going to begin to see them again. From what we're seeing here. Oh, look at all those white, yellow sponges. They're more Primnoa here. See, now we have some. And I think it is competition for space really could be something that drives this a little bit. Yeah, and this is 4 degrees. So do you want to talk a little bit about that, Dave? About the depth and temperature?
So in this particular area, we have two major water masses, and there's one that comes from the Arctic called the Labrador Current, which is really cold. It's actually below zero, and it doesn't freeze to solid because it's salt water. And then below that, there's water that comes in underneath from the Atlantic, and it's the same water that's maybe off of Newfoundland. So a little bit warmer, 4 degrees, and we're only finding these corals in the warmer water mass and not above it. So we keep a close eye on these cliffs, and if the cliffs are in the cold water, we don't think there's usually going to be corals there. So we look for ones that are deep enough that sit in the Atlantic water mass.
Turning up before the filter, is that it's the ROV, yeah. So sometimes we see these bursts of sediment in the water, and it's coming from us. ROV has thrusters, and there is sediment on this ball, so this can happen. Oh my gosh, yeah, that's a very nice cluster. Yes, and there is a coral. See on the top of this primrose. Oh no, it's not it. Yeah. Oh yeah, redfish. Does everybody see the redfish? So this top, they put on the top here. There is a close to the lasers now. There is a redfish hiding behind it. Yes, please. So one thing that is one thing that we need to keep an eye is we have different objectives during a dive. So we're very happy that we saw the gardens. The corals are here, they're healthy, they're abundant. And then we also want to sample. So we need to decide when we need to stop video surveying and beginning to sample with the arm. Yeah. So this is a nice close-up. Trying to hide. Poor guy. And I should mention, because I think the audio will stop soon, and our conversation in the room. But we're planning on doing a second live streaming of another site called the Sentinel, a little bit. I think it's 100 kilometers north of where we are now, and that's going to happen next week. And so you guys should keep looking at Amundsen Science's social media, and you will find the information about when exactly we plan to do that and what time. It is difficult sometimes for us to plan because with the weather changes, it's sometimes it's difficult. We never know if a dive will happen until we get on site and make an assessment with the ROV team and the captain. So hopefully we're going to have another nice dive at a different site, and it's also going to be very exciting. So stay tuned. So question for Trevor. Question for you then, like thinking of time management. We are at 484. We've been exploring a little bit. Do we have time to go all the way to the top and then come down tempo, or do you think we're pushing? Yeah. Okay. I think it's fine because we're not doing a straight line, right? So, okay, Jen, gonna cut? Oh, yeah. So we just say our final words, then we cut. All right. So we're gonna be ending the live streaming soon. So it's gone. Oh, the audio. Yes, I'm sorry. I'm gonna start again. The audio for the live stream is going to stop in the next few minutes. So just wanted to say thank you to everybody that has been watching. we're very excited, very, very happy here, and it's amazing to be able to share this with you. Like I said before, we're all seeing this for the very first time. So I think that is amazing. And so we're going to be back hopefully next week. So stay tuned with Amundsen Science. And I'll just see if anybody else wants to say anything here in the room. Everybody good? Okay. So thank you so much, and see you next week.
And just to answer that last question really fast, the green lasers are 10 centimeters apart. So it just gives us a sense of scale, everything down there. So we're able to see how big all the corals are. Yeah. But thanks everyone for tuning in to this live stream. We'll still have the video for a little bit, so feel free to keep taking a gander at all the cool sea life we're getting to see. And as Barbara mentioned, we will have another live stream next week. Thanks everyone for tuning in.